yeah, bacteria is one of the three domains of life. And then viruses don't count as a domain of life because they're not alive in a biological sense.
Well,
because they're not
weirdly
cells.
enough, then I don't feel guilty when we take an antiviral and destroy a virus because it's not alive. You're not really killing it.
No, you're
I
just
have no
destroying
guilt.
a bunch of chemicals that kind of hang out together.
That's alarming. Welcome to the Skiffy and Fanty Show's SF at School? ? Is the new show name?
That was a show name in the past.
Was it?
I thought so. Like back in the records from like way in the past. Either that or it's one you proposed.
Have you been doing archival research on us?
Yes, I did. Either that or it's like one you proposed at some point for a topic that maybe never got used, but I liked the name, so I went with it.
Okay, well, perfect. Well, but you have a tagline, and I will shut my mouth.
Le rêve d'une bactérie doit devenir deux bactéries.
Do you speak French?
Oui.
Like, legitimately?
Oui, mais ça fait longtemps. J'ai passé en
Okay, hold on. I'm going to give you something. This is my test. In French, say, help, the monkey stole my pants.
Aidez-moi, les singes m'a volé mes pantalons.
Okay.
That's not exactly right, but
French...
there was an error in there, but that's roughly.
Wait,
are you? It's been a while since I've spoken.
Okay.
Yeah, like yes and no. Like speaking is difficult for me because it's been so long, but I can read very efficiently.
Okay, you are the second Skiffy Fanty member with substantial amount of French background, by the way.
Who is the other?
Julia would be our third, but I don't know that Julia really speaks French. I know a little bit of French. Yeah.
I thought Trish or Paul did.
Paul only speaks Latin and
well, close enough.
Anyway, well, I'm Sean.
I'm Daniel.
And on today's show, I'll be asking questions of Daniel about microbiology and discussing how it fits or not in science fiction, fantasy, and horror. And then together, we're going to brainstorm all the amazing stories. Well, maybe not all of them, but perhaps a few.
But first, a reminder to please share your questions and comments with us about the shows at skiffyandfanty.com/listener suggestions. They might get discussed right here on future shows. And if you aren't already, join us on Twitch for Sean's Alphabet streams to be a part of our interactive live streaming format. Finally, be sure to join our Patreon and be part of our special Patreon-only events on Discord. It only takes $1 a month, and you can choose what bad movies you want to torture us with, what good movies you want to watch with us, what books you want to talk about, and all those wonderful things.
Yeah, you even get exclusive podcasts, poll rights. There's a monthly What's It that goes out that lets you know all the things the Skiffy and Fanty Show is up to. And all that. And we use all of that money, I will just note, to pay for hosting the webpage and our files, covering website maintenance, and occasionally replacing broken technology. This is, I want to emphasize, a fan-run show. We have no paid producers or contributors on this show. Not that I wouldn't like to pay, but we just simply don't have the money to do that, and I'm not rich. So this is a show by fans and for fans.
Yeah, we're academics. None of us are rich.
We definitely have no money.
So many
we made
of us,
a bad career choice.
So also, please tell folks about the show and review us on iTunes, Podchaser, Good Pods, and more. Word of mouth is a really big deal for fan-run podcasts, and we need your help to find new ears for this ridiculously awesome show.
Perfect. So, Daniel, before we get to the many, many questions that I have, including questions by some folks in the audience and some that I came up with, I think it would be useful for folks that may not know you to say a little bit about your background, because you are actually a legit scientist.
Yes, so I am a microbiologist. I Though, oddly enough, I did not take any kind of microbiology until I was in grad school. So I went to college my school didn't really have majors at the time. We had our programs of emphasis that we were able to kind of design, or you could take kind of pre-made kind of ones. So I did biochemistry because I was interested in chemistry, and at the time, like high school and younger, I wanted to be like MacGyver. I think I mentioned on an earlier podcast. And so I got into chemistry and did biochem, which was in the chem department. But then as time went on, I got more and more interested in biology. And by just the chance of finding somebody to do research with that kind of meshed with what I was interested in doing, I got into a lab that worked on yeast. So in some ways, that was my start in microbiology because yeast are primarily microscopic. But I didn't really learn the concepts of microbiology and about like bacteria and viruses and such until later in grad school. So I went to college at Juniata College in Pennsylvania, then went to grad school at Washington University in St. Louis, and I did rotations in different kinds of labs that focused on like molecular biology, cell biology, biochemistry, and I ended up settling on a lab, and I did, a combination of molecular, cell, genetics, but in bacteria. And so at that point, really, I began to learn a lot more about microbiology proper.
Interesting. Okay. So I have to ask you some very important questions for people that may not know anything about this field.
Yep.
So these are the most important questions I will ask today.
Okay.
So what is microbiology?
That is a wonderful question that has a hard, a not easy answer to give. So I could probably spend like the whole entire show talking about that. So the simple thing it'd be the study of organisms that are invisible to the naked eye, right? It's the study of microscopic organisms. But you get into some kind of difficulties because, one, you might really not consider viruses to be alive because they're not cell-based, they're not cellular, and therefore they are not biological, they're not alive. They're part of biology, but they're not cells, they're not cellular-based, so they're not alive. You also run into problems that, for historical reasons, things like prions or prions, depending on your pronunciation, which are infectious proteins. They get lumped into microbiology, but it's mostly historical reasons why. Then on the other end of the spectrum, you run into the problem that not all things considered microbiological are actually microbe. They're not microbial in size. So some bacteria actually are really large and you can see them with the naked eye. So technically then they're not microscopic. You have some bacteria that, for instance, are as large as a fruit fly's eye. And even some that have recently been found, I think, that are even bigger. So there's some bacteria that are much larger than like a eukaryotic cell. You also run into the problem that like fungus, because yeast are microscopic and yeast are fungus, but so's like a mushroom. And it's the largest organism on Earth is a mushroom. And so microbiology in some ways includes the largest thing on Earth and the very smallest thing on Earth. And then you have all of like the parasites that are eukaryotic, and so they're not even bacteria or archaea, but they're still in microbiology.
This gets to my next question, which then is, why is microbiology? I'm so confused right now. This sounds like we just threw a bunch of stuff in this field and called it microbiology. So why is it like this, Daniel?
Because it grew from older times before we knew about the invisible things. So all of biology is kind of saddled with you built a system of classification before you knew of what the majority of life on Earth actually was. And so that complicates things when you then discover that everything you knew about and classified is actually like only like a little infinitesimal drop in the actual biology that's out there. And so as you began to realize what things really are, and that like fungus are not plants, they're nothing like plants. In fact, fungus is the most closely related thing to animals. So we're like way closer to fungus than we are to most other groups. Once you realize that, once you're able to sequence DNA and realize that, it becomes problematic for the historical grouping of things. But going back to why, before we even knew what was doing it, we cared because of things like cuisine, preserving food, fermentation, and of course disease. Even though they're the minority, there are bacteria, there's viruses that cause disease, there's fungus that cause disease, there's protists, parasites that cause disease. And so we knew about the disease. Prions kill you. Although those we didn't figure out till like the 70s. But we knew that there were things killing us. And so once we discovered that they were like little tiny cells, that was really the birth of the science of studying the microbial things, once the microscope was invented. So key to microbiology, of why microbiology, would be humans invented the microscope and realized that there's this entire world there that we didn't know about. Other than indirectly, or like you see bacteria on like your shower head, or you see bacteria on your teeth when you wipe off plaque. That's just bacteria. And so like we know they're there, but we didn't really know what they were before we had a microscope.
Oh, okay. So Paul in chat wants you to define a eukaryotic cell for me, but I am pulling out my high school biology. And if I am correct, and Daniel will correct me if I'm wrong, eukaryotic cells refer to basically all animals, plants, things of that nature, things with a cell wall or
no
surrounding a nucleus.
A nucleus. That's the historical definition. So eukaryotes would include. Eukaryotic diversity is actually huge, but it includes things like fungus, algae, the green algae, which gave rise to plants. The fungus gave rise to animals.
Us.
And then included. And then you have like a bunch of other groups of eukaryotic, mostly microbial. Some get big, like brown algae, for instance, or is a different group. Things like paramecium, amoeba, like all those are eukaryotes. But the defining characteristic of a eukaryotic cell has been that eukaryotes have a nucleus, which is a membrane-bound compartment where their DNA is in it. Again, the problem is that we've discovered a bacteria with a membrane-bound organelle that has DNA in it. So we found something that is defined as being not a eukaryote that meets the definition of what a eukaryote is.
And bacteria would typically fall under the other classification, which is prokaryotes.
Yes, the term prokaryote, most bacteriologists, at least I fall on this, that's a term that probably shouldn't get used anymore. It's a little antiquated. In that, in the 1970s, a microbiologist discovered that a lot of the things we thought were bacteria are actually utterly different than bacteria and utterly different from eukaryotes. And so that got named the archaea. So the current understanding of the divisions of life are that there's 3 domains. There's the eukaryotes, there's the archaea, and there are the prokaryotes. Oh God, now I'm saying it. And there are the bacteria. So bacteria are the original that evolved.
Is one of the 3 categories.
Bacteria is one of the 3 domains of life. And then viruses don't count as a domain of life because they're not alive in a biological sense. Because they're not
weirdly
cells.
Enough, then I don't feel guilty when we take an antiviral and destroy a virus because it's not alive. You're not really killing it.
No, you're just destroying a bunch of chemicals that kind of hang out together.
That's alarming. Okay, perfect.
And
I've
got selfish, yeah.
Selfish. Okay, so we got the what and the why. Now you need to know when. When is microbiology, Daniel?
Billions of years ago, in terms of reality of when these things evolved. Bacteria evolved billions of years ago. If Earth's history were a 24-hour clock, I forget when bacteria would be on that clock, but it would be fairly early on. Humans show up at like 11:59 and 50 seconds or something like that. Like we've been around for 10 seconds, if that, in terms of actual life on Earth or the history of the Earth. Most life has been bacterial.
We're
and even animals didn't show up till much later. So yeah,
Yeah, and
bacteria
of
evolved
course,
long
yeah,
ago. And
yeah, and then if you're
then the
thinking in
archaea
human terms,
evolved, and
right?
then
Like,
eukaryotes from
yeah,
archaea.
like all of the like all of the vaguely humanoid species are a blip in that grand scheme because it's over the course of a handful of
Yes,
millions of years.
pretty much.
You know, yeah, okay, that's
Yeah.
bananas. All right,
And then
that
in
gets us to where
the study of microbiology, of course, it was the invention of the microscope. God, I should know the century, but I can't. Late 1700s, 1600s?
okay,
Robert Hooke, whenever he was, you have to Google it. I can't remember the actual even century. But Robert Hooke, an English scientist, invented the microscope and discovered microbes. Later, a Dutch lens grinder named Van Leeuwenhoek invented a lens for the microscope that was much better at resolving to see higher magnification with detail, and he's thought to be the first that could actually see bacteria size of things, the general size of bacteria. But it wasn't until the late 1800s, really, where you began to have the start of bacteriology, of studying them, beginning to really show scientifically that they were the cause of disease. You have the germ theory of disease being born. It's around the same time that Pasteur, he was a key player in it. Pasteur was discovering that, or doing the experiments that really definitively show that there's no spontaneous generation. And then it wasn't until late 1910s, 1920s, when some of the same people that were involved in the development of the nuclear program, actually eventually, were involved in the invention of the electron microscope, that we then had the ability to magnify things up enough to really be able to see viruses and definitively begin to know what they were. Then it wasn't until like Watson and Crick, Rosalind Franklin, the discovery of this structure of DNA and being able to do stuff with DNA, that we really got a hold on things. So, and then not till the 70s that we realized there's this whole entire domain out there that's nothing like bacteria, nothing like eukaryotes, completely new. Who would have thunk it?
That sounds complicated. Okay, so that's
Yes. So sorry.
when. Yeah, no, now
Yes.
we're on to where. Where is microbiology?
Everywhere. As far as we have drilled into the earth, we have found microbes. We have found bacteria living in solid rock, because solid rock still has space in it with like water droplets and channels. And as high up as we've gone into the atmosphere, we have found microbes. Clouds are filled with microbes. They are actually involved, I just lectured on this today, bacteria are involved in nucleating cloud formation, and they are largely responsible for precipitation on our planet. And so we would not necessarily be having snow or rain if it weren't for the bacteria seeding the clouds. At least not to the levels of precipitation that we do have.
That's wild.
And there are reports of finding bacteria growing on the outside of the International Space Station, and it's unclear if they came from an extraterrestrial source or, the components got contaminated as they got up into space, or if things are just getting out of our atmosphere and happening to then seed and start to grow.
That's wild. Okay, so then there's a question in chat, which is somewhat said in jest, but I think you would agree with this, that Triffity Matt says, so rainwater isn't pure after all. And I'm guessing, Daniel, you would probably say pretty much any water outside is not pure.
Correct, yeah.
Yeah.
Ultra filtered, but even, filtered water should be sterilized. But
what about Brita filters?
That's why for most of human history, people drank alcohol, not water, because water was deadly. Water will kill you. Yeah. So
does that mean like your Brita filter then also doesn't actually clear? It doesn't come anywhere close to clearing any of it?
No, it should filter. Yeah.
All of it?
I don't know the size of Brita filter, but when I sterilize things in lab through filters, they'll block out most cells unless you have weirdo like cells that are the size of normal viruses, like nanometer in size. Okay. They do exist. They're just not common.
Okay.
So, yes, they'll filter out and it'll stay sterile forever, in theory.
In theory, yeah.
So, okay, question. You can filter these things out.
Yeah. So, how is microbiology?
How?
Yeah, how, Daniel? These are the pressing questions that people want to know.
how can I interpret that? How microbiology, because cells evolved, I guess, and what microbes are and what cells are little containers of organized like metabolism, of biochemical reactions. So like viruses in some ways are like molecules and stuff doing reactions out in the world and need to be in a cell. But somehow cells evolved in that chemical reactions got caught up inside something that's capable of growing and dividing into two. Cell division. And so it's a self-perpetuating system of biochemical reactions. That's kind of what life is. So that's how, if that was the question. Yeah.
That's interesting.
But
So
my
I know
quote
this
at the
is
beginning is from a bacteriologist. It means in French, the dream of a bacteria must be to become two bacteria. And that's really what cell biology is about, that cells, they exist to grow and divide to make more cells.
Okay, so I know this is an obvious question because I think I've asked this of you before, but I'm going to ask it again anyway because I don't think I asked you in public. Based on my admittedly layman's understanding of bacteria, which is a thing that you do study, life as we know it in this planet pretty much can't exist if bacteria doesn't exist.
Correct. A few different ways. We need biological elements, right? We need carbon, we need nitrogen, we need phosphorus, we need things that make up our cells. They're responsible for cycling those. The only thing capable on Earth of doing nitrogen fixation, other than humans invented a way to do it artificially, which has destroyed our planet, but bacteria are the only thing that can naturally fix nitrogen. So they are key. If we lost them, we would lose the ability, life would lose the ability to incorporate nitrogen into new cells. So as things decompose and you get nitrogen gas back into the atmosphere, even without decomposition, it goes back in the atmosphere, there'd be no way to capture it back into molecules that life could use. The other way is that no animal or plant would exist. Pretty much all eukaryotes would just cease to function because everything relies on bacteria inside of them to exist. For us, for mammals, we're just really, really sick if we don't have bacteria, and you likely die from something very quickly because you wouldn't have a developed immune system. But most animal species, they actually fail to develop into a mature organism like in utero or in early stages of development. Like it just doesn't happen right if microbes aren't present in them.
Oh. Well, and even, I mean, I suspect a lot of people know this, but it comes up so often, even in my doctor visits occasionally, where just the sheer number of bacteria that my body relies on for basic functions. And if you take them out, like suddenly I'm dead, basically. At some point I die. Right,
And
digestion, as Triffid is pointing out, right, would be, I would argue, probably impossible, right, without bacteria. I mean,
we have our own enzymes to do it, but we wouldn't be getting the same things out. Yeah, you need them for complete digestion, at least, for getting all nutrients. It would screw things up really badly.
We would die, I imagine.
Well, again,
can we survive without
completely germ-free? A human would not immediately die.
Well, I don't mean like you just keel over, right? Like instant, like you took my bacteria,
yes, most
Daniel.
likely, yes, yeah. You'd have to be a bubble person, effectively. And you could still eat, you could still digest, you'd just be very sickly and you wouldn't be getting the nutrients you normally would.
What else would you have other problems as a consequence of, like, if you could be born as a human without any bacteria, like let's say that's just possible, and you're living in a bubble, right? If you're exposed to any bacteria and your body's never been exposed before, what are the consequences at that point?
You die. Your immune
body
system
has
would
no
just like go
relationship.
crazy and your immune system would kill you.
Whoa.
It would be improperly developed, or it's so improperly developed that it just doesn't put up anything and the bacteria grow and overtake you, whereas normally the immune system would keep them in check. So they'd like eat you from the inside out, I guess.
Would that even be the case for, like, because we live in, I don't know if symbiosis is the correct word, but we're in some sort of symbiotic relationship with certain bacteria. That would include, they would just come in and like you'd end up dying.
Yeah, so symbiosis can be a general term for any kind of like interspecies interaction that's very close.
Okay.
And you could use it to both refer to beneficial ones where like both species benefit, that's mutualism. You could have interactions where one benefits, one doesn't really get affected one way or the other. That's called commensalism. And it's not even certain. It might just be that one is affected. We just don't know how it's affected, or it's a very subtle effect. Then you have something called amensalism, which one of my colleagues calls assholism, because it's when one species is not helped or bothered, but you fuck over the other species as a result. So the interaction hurts somebody else, hurts another species, but it doesn't even benefit you. So you're screwing them over with no benefit to yourself. So that's amensalism. And then you have things like parasitism, predation, things where you benefit and you hurt something else in the process.
Okay, gotcha.
So those
okay,
are the possibilities great. Of symbiosis. But pretty much all communities and like any organism, most organism on Earth is actually really a community of different organisms, because you even have cases where like bacteria are filled with viruses. So if you consider a virus part of a community, it is. You have bacteria living in bacteria. You have bacteria living in viruses actually have been found. You have viruses inside viruses. It's almost like biology. Most of it is like Russian nesting dolls in terms of cells and cells. A dinoflagellate would be an example of that, where they have, I think it's like or even termite. Termites can digest wood because there's bacteria in a protist that's in the termite gut. So you have many situations like that where you have cells and cells and cells and cells. And that gets back to like whatever the question was of can we live without them? No, because I would consider mitochondria to actually be bacteria. Mitochondria have a bacterial origin in evolution. They were once free-living bacteria. They were just captured and enslaved. And so I am an advocate for freedom for the mitochondrial slaves, but that would lead to my death. Similarly, chloroplasts, plants don't do photosynthesis. Bacteria do photosynthesis. Algae don't do photosynthesis. Chloroplasts do photosynthesis, and their chloroplasts are bacteria.
Literally, the second you said the word chloroplasts, Paul asked that question in chat, like instantaneously from my end. I thought that was very funny. So, okay, so there's a bunch of other questions. I'm sure there'll be more that'll come up in chat, but there were a couple that I don't even understand, and so I'm sure you will help me with these. But given that we keep bringing up viruses and bacteria, there's a question here from, I believe, Paul, which is about the evolutionary relationship between viruses and bacteria. And the questions are there bacteria that act like viruses? And then the second part of that is, what do we know about the evolution of viruses in the first place? So I know that's kind of big. So give me, I guess, the Cliff Notes versions of how you would approach those. Yeah.
In terms of the relationship, the thought is that there's different theories. One is that cells may have evolved from viruses, and the other is that viruses may have evolved from cells. And probably it's gone both ways, because there's some evidence of viruses, that we've discovered that look like cells. They have membranes. They have like all kinds of genes that you would find in a cell, genes that viruses have no need for, like doing photosynthesis, for instance. And so it's thought it was a cell that through evolution lost a bunch of its function, and it can no longer grow, it can no longer divide. If you can't do that, you're not really a cell anymore. You can't grow, you can't make more of yourself. You need something else to live in to be able to do that process. And so it's thought that some cells degenerated into viruses. On the other hand, some viruses may have, and there's evidence of viruses that infect bacteria that steal membrane to make little houses for themselves to replicate. Well, now you have DNA replicating inside of a little membrane house that's a nucleus. And so cells may have evolved from viruses doing their thing. So other times, like bits of chemical machinery may have escaped a cell and become a virus. So it's probably been a two-way street in terms of the evolution.
But hold on, Daniel. Are you suggesting that one of these theories may be that ultimately humans are a virus?
It depends what you mean by virus. Not a virus in a biological sense, but in a functional sense, an argument could be made, yes.
Okay, great. And then I guess that other question, which is the one about, are there bacteria that act like viruses? And I would actually ask if you could explain what the act like viruses really means in this context.
Yeah, so that gets back to like my example of the chloroplast of the mitochondria, that they're really just bacteria. They're called something else for historical reasons because we didn't know about bacteria when we discovered the mitochondria and the chloroplast. And we didn't know that evolutionary genetic relationship. There are countless bacterial species out there that need a host cell to live in. The bacteria cannot exist apart from the host cell. In some ways, that's the definition of viruses, right? Things that can't live outside of a host cell. These are still those cells because they can grow and divide inside of the host only. Like mitochondria grow and divide inside of our cells, or chloroplasts do in plant cells or algae. And so an example of that would be the Rickettsia bacteria. They're thought to be close evolutionarily to what gave rise to the mitochondria. Rickettsia causes typhus, and it also Rocky Mountain spotted fever, I think is the term for it. So the Rickettsia cause disease in mammals, and they're viral in some regards in that they don't exist outside of host cell. They can only grow and divide inside the host, but rather than helping, like mitochondria do by giving us energy, they actually hurt the host in the process. So that's very virus-like.
Oh my goodness. So I had to Google it really quick because if I'm correct, it's a flea-borne disease.
Yes. Yeah.
Oh, that's terrifying. Okay.
It's not a pleasant disease. It's not a pleasant bacteria to study because you need, host cells to grow it in too. So.
Does that, when you say unpleasant, I mean, to me, the host cells suggest that it's not always going to be, lower ends of the life forms, things where we don't necessarily feel much guilt about it being harmed. You're thinking possibly infecting.
No, I just mean like technically it's difficult to work with.
Oh, okay.
Because it's harder to work with in terms of like resources that you have and the amount of time to grow them and all that kind of stuff.
Oh, okay. You didn't mean ethically.
That's what I mean. No. Fuck them.
Okay. Okay, well then, Triffity Mats in chat was wondering if you could talk a little bit about phages, which I suppose a part of that question is also explaining for everyone what the hell that even is, because my guess is most of us have never learned the word phage. So what is that?
Well, you'll have to ask Prince Justin, although I guess, I don't know, is phagus, maybe that's Greek, not Latin, I guess, actually, never mind. So phage means to eat.
To eat.
Yeah, so like a macrophage is an immune cell we have. It means a big eater. It's an immune cell that eats bacteria that invade us and breaks them down. It's like a cell that digests it. So effectively, it's like an amoeba in the environment, right? It preys on bacteria.
They literally called it Big Eater. That's the name they gave it. Big Eater.
Macrophage. Yes, a macrophage is just big eater.
That's so stupid.
So, a phage is historically the term that was given to viruses that infect bacteria. Because they were discovered by, like, if you plate bacteria on an agar plate and you get a lawn of bacteria growing, which means the whole plate is just covered with bacterial cells growing in, like, their little mucoidy kind of growth. If some of them are infected by the virus kills them and they burst. And so you have little circles of clearing. And so they were like, something ate those cells because there's no more cells there. There's empty space, which they called a plaque. So there's these plaques. We're going to name what the thing is that destroyed them a phage. And so they became known as bacteriophage. And then they were actually some of the first kind of viruses to be discovered and studied before we really knew what viruses were. Because the term virus just actually derives from the term poison. So a virus literally means a poison.
Okay, I have to follow that up.
The same as the term germ just means seed, because you had the theory that germs came from a seed of something really tiny that gave rise to illness, like a seed, like a germ line cell of the germ line of wheat germ, for instance. The germ is referring to a seed that grows to give rise to the thing you see. So that's the history of that terminology.
So with viruses, then, I'm guessing they're not by default always harmful, or are they always harmful?
No,
they're not. Okay, explain.
They're not. So one, there's huge numbers. I forget the counts, but there's an uncountable number that pass through us all the time. They're very species specific usually, so it's only certain, like if you have a virus for a bacteria, there's no way it's going to infect a human cell, for instance. Even between two species of bacteria that are fairly closely related, the virus will usually be specific only for the one species, not the other. Once you get to animal viruses, there's some, influenza is good at infecting birds and humans and pigs. So there are some examples that have a fairly broad range, but most are very specific towards things. They also might be specific to only certain cell types that they infect. Other viruses, though, will get into a host, but they've evolved to not really cause that much damage. Like the common cold in us, it's an inconvenience, it's an annoyance, but it's not really harmful. At least not on the broad spectrum of our entire bodies, even if a few cells are hurt. But then some viruses actually protect their host. There are examples of beneficial viruses. There's, for instance, a virus that's in a fungus that associates with plants that now gives the plant ability to survive in drought conditions and during conditions of metal toxicity, I believe, has been studied before. It's thought that the ability to survive freezing conditions in some species that live in the Antarctic, like fish and such, or small macroinvertebrates that are in the ocean there. It's thought that some of that comes from symbiosis with bacteria and viruses that are present in them that now gives them the ability to survive in harsh conditions. Viruses can help protect a lot of insects from other kinds of diseases. There's bacteria that can protect insects from viral diseases. So there's lots of beneficial relationships, including with viruses. And also viruses are beneficial ecosystem-wise in that they are helping recycling of things, breaking things down. Without them eating all the bacteria, for instance, we'd be drowning in bacteria. So they help recycle stuff.
Okay. Well, that's pretty cool. All right.
And yeah,
so
just in a little bit of seawater, there's more bacteria, or there's more viral particles than there are stars in the observable universe.
Good Lord. It's a lot.
There's a lot of viruses, yeah.
All right, I have to ask you a really tough question. This one's very near and dear to my heart because I Googled you and then I found this. And I'm curious. So currently, at least according to your university webpage, you have a paper in peer review which you've co-written and researched with several other authors, and it is entitled Bacteriophage SP01 Gene Product 56 GP56 inhibits Bacillus subtilis cell division by interacting with div-IC FTS cell to prevent PBP2B FTSW recruitment. And I wanted, if you could, to translate that into English for those of us that are not scientists. What the hell is this about?
Apparently that needs to be updated because it's been published for a while now.
Okay, so it is out there.
Am I getting tenure even? Oh yeah, it's out there. It was on bioRxiv and then it's been published in Journal of Bacteriology. So bacteriophage, we already went over. It means bacterial virus. So there's a bacterial virus that's name is SP01. It's just the name of the virus. Okay. It has a gene in it that makes a protein that's called protein 56, gene product 56. So it makes a protein, and that protein inhibits cell division of a gram-positive bacteria called Bacillus subtilis, which is the Latin name for it's a common soil bacteria. It is related to species that are used for fermentation, particularly in Asian cuisine. For instance, if you've ever had natto, the Japanese delicacy, it is fermented soybeans. It is made by a Bacillus natto species. Bacillus subtilis is harmless to us. Some bacillus are beneficial. But Bacillus anthracis is probably the most well-known because it's the one that causes anthrax. In any case, this bacterial virus makes a protein that inhibits cell division of this bacteria. And it does that by interacting with two components of a complex of proteins, and their names are Div1C and FTSL, that are involved in cell division. And the way that it inhibits, the mechanism, is that it prevents the recruitment of enzymes to that place where the cell needs to divide into two. It prevents these two enzymes from coming that are necessary to build new cell wall. So bacteria, like you had mentioned before, Sean, cell walls, most everything except animals has cell walls or some kind of form of cell wall. Bacteria have cell walls, and so when bacterial cells divide, they not only have to constrict their membrane, they need to put down new cell wall crosswise. And if you don't do that, you can't divide. And so what this phage does, this virus does, is that it makes a protein that goes and localizes to where the cell is trying to divide and screws up the ability of the cell to make the cell wall. And those weird letters and numbers are just the names of the proteins that are involved.
You should be a science educator. Like what you just said,
did that actually make sense?
Hey, what you just told me, everything you just explained, made this thing that seemed incomprehensible to me. Like, that's super easy. Now I get it. Like, oh yeah, that's like basic. Like you made it seem really simple. So.
Well, I mean, most of it
bonus points.
It's just, it's occluded by all this terminology and such, right? So
yeah. Can I ask then? So you mentioned the anthrax bacteria. Is part of this study then about trying to figure out ways that would allow us to combat more harmful forms of this particular family of bacteria? Is that what the goal is, or is this just okay?
It's not the immediate goal. The goal is really just to understand the basic science of how this is occurring. But the main protein in this process of cell division, is actually a potential target that people investigate, that other labs investigate as a potential new target for antibiotic development, for instance. Because this process that they do division with, it's unique enough to bacteria that a drug would be affecting bacteria, but wouldn't be affecting our cells, wouldn't even be affecting our mitochondria, unlike some antibiotics that we take. In addition, it might be, different enough between bacterial species that you could maybe make an antibiotic that only affected one as opposed to many. And then also the idea that it might be more beneficial to try to kill bacteria with viruses rather than chemicals. Because a chemical can evolve, a virus can evolve. So if bacteria evolve resistance to chemicals, you're screwed, the chemical's useless. If bacteria evolve resistance to a virus, the virus can evolve ways to now one-up the bacteria. They're in an arms race. And so that's a little more potentially effective than just trying to kill things with a chemical that's utterly benign and inert.
Kind of the way I'm viewing this as somebody from the outside is almost like you're bringing to antibacterial materials the same methodology we now use for dealing with antivirals and vaccines, which is it is an arms race trying to stop the flu, for example. We can't really cure the flu, right? So so it's kind of the same thing. Why don't we use
viruses to do the arms race rather than us trying to constantly try to do things? Yeah,
that is so fun. Okay. So there's that. So your research has kind of contributed to this larger conversation about different ways we can interact with these bacteria and find ways of dealing with it. Okay.
Right. And that's really what most research is. And most of the advances that have come to benefit medicine and society have not come from research into the topic. For instance, CRISPR-Cas that was discovered to do gene editing now, that has the potential to do a lot of good, was discovered just by studying like why bacteria are able to resist viral infections. Absolutely nothing to do with medicine, nothing to do with humans. But hugely, way more impactful on society now than like all the billions of dollars we spend on cancer research. And so, yes, directed research into studying something has its benefits, but a part of me always cringes when somebody talks about, donating their money to cancer research, doing this, because it's an easy thing to get behind, but in terms of actual benefits and productivity, it's not what gives the biggest gains to humans. Like, if you look at the record of where most of the huge developments have come, they've come from not studying medicine. They've come from studying basic general biology. And then somebody realized, oh, wow, this could be applied to this. And then people did the directed research. And so actual discovery research has no business being applied to a specific disease or problem. We should be doing discovery research on just general biology to discover things that then others can apply.
Interesting. Okay, this is exciting. Well, thank you, Daniel. I have learned more about what you do.
But that's my bias.
No,
but yeah, we should probably talk about like sci-fi or something. We're like,
sure, yeah. Well, I do have one last question that is, I guess, kind of sci-fi, but is maybe a little bit more like very near future-y type sci-fi. But like, obviously, bacteria, single-celled life forms, any sort of life form that is more tiny, microbial, smallish, is part of a huge conversation about life outside of our planet. You'd already mentioned there may be some stories or suggestion that possibly bacteria has grown outside of the International Space Station. And so that kind of easily leads us into a question I have, which is the fact that we are looking for microbial life of some variety on other planets. Mars is like the big one, but even like Europa and some of the other moons and trying to see that. And so I'm curious, from your view as a scientist, why do you think this is the most promising focus for extraterrestrial life?
Because they're the simplest life in being just a cell. And so if we are going to say and use, it's somewhat arbitrary, but if we're going to arbitrarily define life as being cellular based, then just having a cell on its own doing things is effectively a bacteria. And so, or something bacterial-like, or an archaea. And so I'd expect the first thing to evolve, if you're having the evolution of a cell, it's going to be an archaea or a bacteria or something like that. It would probably get another name because it would have a distinct evolutionary origin, and have different genetics to it, but it would be functionally like a bacteria or an archaea. And so that's what you'd expect to find out there. And also, from what we know on Earth, it's the archaea and the bacteria that live in the widest range of conditions, from, what we consider normal to the most inhospitable environments. There are archaea that live in acid mine drainage that has negative pH, where if you put a metal shovel into it, the shovel would dissolve in seconds. And there are like cells living in that and thriving. And so adverse conditions, what we would consider inhospitable to life, even the lack of water. There's cells on Earth that are like, I don't care. I'm just going to keep doing my thing, or I'm going to hang out until you give me some water and then I'll do my thing. And so we'd expect that would be similar on other planetary bodies. If chemistry evolved in a cellular context, that's what it would look like. And we have examples of that here on Earth, in parts of the Earth that are conditions that are very alien-like or extraterrestrial-like in extremes.
Okay, so I'm going to ask a question that you're answering as a scientist, but you're not answering, like, the answer you give me is not like a science answer, but like on a scale from 1 to 10, where 10 is like absolutely guaranteed 100% and 1 is like absolutely not, never will happen. what do you think the likelihood that there is currently living some form of microbial life somewhere other than this planet in our solar system?
Like 9.5, 9.8.
That's really confident, Daniel.
Yeah.
Well, why? Why that confidence? Yeah.
Just, I mean, to some degree, I should know this, but what's that, the sort of extraterrestrial intelligence? What is the calculation called?
Oh,
Why am I blanking on?
it's Drake equation.
Drake equation, yeah. If you do that on the microbial sense, it becomes like near certainty of like not a civilization like human-like, but just like cellular life. If you tried to estimate that, I imagine you're going to get a number that's really, really likely. Again, also just because we see in the continuum from non-life to life, we see things that are sort of in between. And so the idea that it never happened anywhere else in the entire cosmos is kind of absurd. The other would be that there has been evidence of life on other planets, or other bodies, including the moon, or Mars, excuse me. I don't remember the decade. It may have been the 70s, but I remember going to a talk at an ASM meeting where a NASA biologist talked about how in the 70s, during the various missions, they designed technology to take samples to see whether there was signs of life, and all the things that they got back were affirmative and positive for the signs of microbial life, doing metabolism and changing elements like they do here on Earth. But NASA basically then, after they got the results, they're like, oh, okay, that's cool, but we're going to play it down because this is too big of news. This is too huge to just say and get behind, yes, we discovered life. And so they kind of they made a proposal, they approved it as being evidence for signs of life, but then when they got the positive result, they got cold feet and never really did anything with it.
That's alarming.
So that's, I mean, in some ways that's hearsay because it was like somebody's talk at a meeting that I heard, and I might be remembering things differently as well. But that's the general sense that I got, that NASA has done experiments specifically designed to test for extraterrestrial life and gotten unequivocal positive results, but then out of abundance of caution has downplayed it or said, well, we can keep looking. You
can't entirely blame them, right?
So,
because
no, I don't. Yeah. I don't. But anywhere in science, if you make a huge claim like that, you need really good evidence, right? But
well, and of course, if you know how the media works, like they would say, like, "Oh, we found some single-celled simple organism," and then they'll be like, "Oh, it was like little green men, and there's a Martian civilization, and it lives in the face." Like, yeah, because every single time, like there was a story, I don't know, a month ago, they detected a signal. The scientists hadn't even said; they just said it wasn't from our solar system, and all these news articles came out like they might have found alien life, and like they never even said the word alien life. They just said it wasn't from our solar system. That's not the same thing.
Yeah.
Anyway.
No, I totally do not blame them and such. I seem to recall the scientists being somewhat bitter that they didn't follow up and do further experiments to try to repeat it and such, and went to do like other kinds of tests afterwards. So anyway.
Okay, sci-fi questions. I got a bunch of them.
Yes.
Let's see what we can do. So
all right, we can
in terms of science
fiction. And then
fiction,
if people are interested, we can always do a part 2, I guess. That's just on the speculation.
This is pretty big. So writing. So when you're reading science fiction, which you do, what is something that you would like to see writers do with presenting, dealing with, exploring basically microbial life, bacteria, or whatever? What is something you'd like to see in science fiction stories that you maybe don't?
I mean, quite simply, just to see them included, because they affect every single process you can possibly think of on the planet. And so whatever you're focusing on in your sci-fi story, microbes could play a part in it. Of whether it's, elemental nutrient cycling on the planet, the climate, precipitation, as I mentioned before. the cuisine of the planet, right? If you have a planet, are they using microbes to do fermentation? Are they to help prevent food spoilage? Is that even an issue? When you consume food, or consume anything, when you breathe, you're breathing in microbes, right? They're covering you. If a species comes here, we go there, we bring the microbes. What happens when the two meet, etc. We always talk about first contact on the human-like alien terms. What about first contact amongst the microbes that are living on those aliens? What kind of possible things could happen? The possibilities are endless. So I guess the simple answer, again, would just be, like, include them. Because they're damned important. And they're ignored always.
So related to that question, because you kind of brought it up, if you're exploring the cosmos, as it were, and you go to some alien world or whatever, right? Naturally, you're going to interact, if that world has life, some form of microbial life. And so I was curious about, if you're on an alien world, and based on your knowledge of microbiology from our planet, what might be the threats that exist there for us as humans going to some alien planet with life on it? What might be the concerns we should have if we're doing that?
So I guess the biggest concerns is how our bodies would react to them. And so our bodies, our cells, capable molecularly of recognizing the presence of those aliens if they happen to get in us or on us. If they are, it might just be, as long as they don't get into a wound or something like that, or you don't eat them, it might not be a huge problem. So it really comes down to the complexity of what happens on the first contact. Do you have, does that alien microbe actually begin to grow and divide? You might start getting a problem if a bunch of them grow. One, if they just keep growing and we don't recognize them, they could grow to really high population and start screwing us up that way. But then also, if we do detect them, our immune system might go absolutely apeshit, particularly if it's something really alien in the sense of our molecules, they have molecules or things that we detect, but are so different that you just attack, but you end up doing collateral damage to yourself in the process. And so more than likely, I would think it would be a complex and a variable reaction. And there's something to be said for that. The most likely is that there's not going to be a problem, in that most pathogens have evolved to deal. So when a pathogen gets inside of us, for instance, they cause disease usually because they're trying to get out of us. They don't want to be in us. We're not their natural host for many of them. So they react to us and our bodies the same way they're reacting in the environment to being attacked by amoeba that are trying to eat them or something. They do things, and in that process, they harm us. The ones, if they're alien, they might not have that reaction at all. They might be completely befuddled, as it were, when they get inside of us, and they might not even sense each other. And so you might have absolutely benign kind of no effect, in a way. So there's lots of possibilities, and it's anyone's guess what would actually happen.
And I suppose as a part of this equation is the opposite direction. This is the thing I was talking with my students about with regards to sending probes and stuff to Mars. If there is bacterial life, if we bring stuff with us there, right, maybe we could be the ones that harm the life that's there. So in the same way, I guess it could go the opposite way. If we're not, I don't even think that sterile even matters for human bodies, because we have bacteria in us. You can't, how do you even begin to keep your,
You can't. No. Other than the human is in a contained suit, right? And so if that were the case, that would be limiting it in theory, but
you poop at some point.
If there were any biological connection, well, that would have to be contained and kept segregated from the other. Yeah.
There's a cool science fiction story to be written there of First Contact, but it's people behind giant glass walls trying to talk to the aliens because we don't. Someone should write that story. So let me ask a slightly different question. So we'll go back to the Earth, right? Time travel. Does as a microbiologist, do time travel stories annoy you? Like I
No.
go back, I go back to 912, the current era, right? And I'm fine. I'm just eating the food like a rat. I'm drinking the beer. I'm like wandering. Everything's fine. Does that bother the crap out of you?
I guess I never really thought about it or haven't read enough time travel where that kind of long, distant past, kind of pre-1850s time, pre-germ theory of disease time travel came into play. In general, time travel always annoyed me more because if you did time travel, you'd also have to do space travel because the Earth is moving. And that always concerned me, like, how the hell are you doing that? As opposed to, mere materializing in the middle of outer space. I guess it annoys me the same way as the fantasy genre will annoy me sometimes in general, of people in these, particularly the Western kind of European medieval times type epic fantasy, where it's clear there's no germ theory of disease going on in this place. Maybe they have magic treating disease, but then how is that magic dealing with microbes? What is actually happening there? Nobody talks about it. But why are people not dying of disease all the time? At that time period in particular, people should just be randomly dying. The hero of your story should accidentally trip and break their ankle and die from the wound from sepsis, or you accidentally get a cut on your hand, or you decide to drink some water out of that stream, and all of a sudden your trusted companion, you're burying them because they're dead of dysentery. There's none of that random chaos that comes from what biology really is. And so I guess that, in general, in fiction, that bothers me sometimes.
That's interesting, because I'm not normally bothered by most of these things when we go back into the past, but it does annoy me knowing that people go back in time and there doesn't seem to be any consideration about the preparation of one's gut flora or the potential diseases that we don't vaccinate for because they don't exist. I mean, if polio is completely eradicated off the face of the earth, right? And in a century, we're not gonna do anything for it. It's not a big deal. So if I send somebody back in time, are we going to some ancient lab where you hang out and being get out your old polio? We got to make some
there's
vaccines.
No vaccine, no antibiotics. Yeah.
Yeah.
I guess the biggest thing that would probably occur, though, mentioning disrupting your gut microbes, is that the people that traveled back because of the food and such, you'd really get GI distressed. And so that's not necessarily a topic you're talking about in the books of people taking a shit, or if all of a sudden you have diarrhea all the time because you've traveled back in time, it's not necessarily something you're writing about in the novel. But that would be most common, I would say. You just completely disrupt your flora. But it would...
That's the thing.
You'd adjust. You'd adjust, with a bit of time and be okay.
Yeah. See, this is on my mind because I've actually traveled to, outside of the United States to parts of the world where
it's
you do have to be a little bit careful.
That.
Yeah.
Yeah.
And so I feel anytime I read a fantasy story where somebody travels back in time, or a sci-fi one where they do the same, and they don't deal with this problem, and I'm just sitting here going, but you've never traveled outside of the West then. You don't know what it's like where you can't drink the water what it's like because you will shit yourself. Yeah. So
right, exactly.
I know. Yeah. So this is a reminder. If you go to a place where the water's not clean and you drink from the tap, even if the water's fine for the locals, you may still shit out your life. I'm just saying.
And it might be bloody when you do it. So
it might be bloody. Yeah, I've actually had to deal with getting antibacterial medication because of I caught my gut flora went just lost its mind because I'd been exposed to some stuff that just sent it just probably too much seafood. But it was just for weeks. right? Where it was just,
Mm-hmm.
it wasn't normal. I was eating bland food and they were just like, we're giving you an
Mm-hmm.
antibiotic and you ran through this in a week and I was fine. But yeah.
That actually, so your question, I just realized something. To some extent, people from today's age traveling back at time would be at a more significant risk of dying from infection from the foods. So, for instance, I just got done reading a biography of Andrew Jackson. And Andrew Jackson, his whole life, he was riddled with GI issues because of parasitic infections, because they were common then. And humans currently have evolved, our internal body temperature has actually decreased. So you may have heard like human body temperature should be 98.6 degrees Fahrenheit or 37 degrees de Celsius. No, but that's no longer normal for humans. It's actually cooler. And the hypothesis is that because we are so ultra hygienic to our detriment in some ways,
Yeah.
we are so parasite free that our bodies have the population has evolved to have a lower body temperature because we don't need to anymore because we're not riddled with parasites or we don't have that risk. But now if you took someone like that and traveled that had an internal body temperature that was a little cooler, they went back to a time when there was no clean drinking water. The food wasn't handled the same way. You're very quickly going to get a parasitic infection, particularly if you go to the south. And as a result, the people who live there in that time period, their body temperatures are accustomed to it. And so they will handle with inconvenience the parasitic infection. You, as time traveler, might not survive that process. And so there would be an increased risk, yeah.
That's wild. Yeah. See? This is why you should study microbiology at least a little bit. You might learn a thing or two. Okay, so I want to ask one last science fiction-y question, which is more a creative one for you. So you're the microbiologist here. If you could create a new alien life form based on your knowledge of biology broadly, what kind of life form would you create and why? You can do anything you want as long as it's vaguely close to science.
So there are species that are capable of alchemy, in the sense that they can make gold through metabolism. Now, it is a metabolism where they're taking something that has gold in it and changing the amount of electrons that it has so that the gold is now in solid, pure gold form. It takes more money to do the process than you would get out in actual gold, so it's not like people are going to do this to get rich. But one of the cool things about microbes, there's a famous ecologist that basically had said, wherever you are, there's bacteria that have evolved to live there, or archaea. 2, if you can invent a molecule, there's a bacteria in archaea that has evolved to degrade and break down that molecule to either get electrons or energy or just the building block of the elements. And so kind of making a bacteria that evolved in a unique environment in some way that was capable of a new kind of metabolism based on an environment we don't have on Earth would be cool. Maybe other kinds of elements in the periodic table. And then also kind of maybe hypothesizing a way where it's not just electrons that they're handling, but maybe there's a situation where they're changing matter in other ways. And maybe you could have a bacteria that was actually capable of moving neutrons or protons in a way that now did transmutation. And so then you could actually have an alchemist bacteria that was able to take lead and change it into gold, for instance. There's probably a better element you could pick, but changing something into gold or doing some kind of cool thing like that. That would be fun.
That is exciting. Cool. Okay. So we may have to do another science-y conversation because there's a lot of territory left, but we don't have time.
There's a lot out. Yeah. If people care and are interested, sure.
Yeah, we could have a lot of fun. So we're going to have to close out the show because otherwise we'll be here for another hour for Daniel's second lecture. So for that, with that in mind, folks, thank you again for being here. If you're live with us, and thanks for listening. If you'd like to let us know what you thought about this episode, or you got other questions that Daniel can answer in email, you can go to skiffingfanny.com/listener suggestions, or send us an email at skiffingfanny@gmail.com. We're always looking for new guests, and we're always looking for books to review and all kinds of fun things. And we always love to hear from folks. So if you have things you'd like us to cover, topics that maybe we've never covered before, or books you'd like us to read, or even movies you'd like us to watch, please send us a message. We'd love to hear it. For places to find us, we're Skiffy and Fanty on pretty much every social media site other than Twitter, because we're not there anymore. So Blue Sky, Threads, Insta, TikTok, Mastodon, a bunch of other places. All of our links, including our newsletter, are at our Linktree, which is linktr.ee/Skiffy and Fanty. And then you can go to patreon.com/Skiffy and Fanty to support the show with all of your hard-earned dollars. And then please do go leave reviews on iTunes and all of those fancy places if you can. As a reminder too, this show is live every Friday at 6:30 p.m. Central on twitch.tv/alphabetstreams, which is where I also stream Tuesdays and Thursdays at 7 p.m. Central. And I'm at SeanDuke.net. I have a Patreon, all that good stuff. My link tree is just /SeanDuke.
You can find me on Blue Sky at Reading 1000 Lives. That's Reading 1000 Lives. And my general reviews at reading1000lives.com, the Skiffy and Fanty blog, and elsewhere.
Yeah. And I believe, if I'm correct, Daniel will be releasing a new paper very soon that is about my favorite bacteria, Schonachus macomicus, which is this brand new bacteria that Daniel has just newly discovered. This bacteria, what exactly does it do, Daniel? It's really cool.
Actually, I think it would most likely, if to name it after you, it would be an archaea that we might have on hand here.
Oh, it's an archaea. So
it's an archaea.
That means,
yes.
So it's really old and sexy.
Pretty much, yes.
Perfect.
It's actually a particular kind of the reason why it's named after you is that it's a part of a group of archaea called the Asgardian archaea. The first member was Loki archaea. There's also a Heimdall archaea, a Odin archaea, and a Thor archaea group. And so we are trying to find out which group exactly of the Asgardian archaea that the SeanDuke variety actually falls into. But they are an ancient lineage, but they are thought to have adapted many of the cool handbag of tricks that eukaryotic cells came to use. So they were instrumental in becoming eukaryotic and allowing all of us to thrive.
You're welcome. Okay, folks. And on that note, awkward ending, and scene.
May the microbes be with you.
If you want to support this show, you can go to patreon.com/SkiffyandFanty or skiffyandfanty.com, our website, where you can get access to all of our fancy things. Our music comes from Holy Mole. You can support him and his work at patreon.com/HolyMole. Thank you for listening.